And one of the biggest problems in the field right now is that while we certainly have technologies that I believe are probably, as effective as exercise, perhaps more effective than exercise to some degree. I mean, mTOR inhibition, senolytics, fecal microbiota transplantation or similar. These things are all very cheap. Anybody can do it. Physicians are prescribing it, and there is next to zero human data for any of this because nobody has any incentive to run the clinical trials at cost. I mean, if if a philanthropist could spend $10 million and answer all the questions I wanted answered here, but nobody's lining up to do this yet.
Hi, everyone. Welcome to episode 11 of The Polling Principle. I'm here today with Reason and Murat, of course, let's say the founders of River Bio, two people that have been doing a lot of really interesting work on longevity for many years. I'm actually very excited to have both of you today. I think this will be a very interesting conversation. And maybe for the sake of intro, we've known each other, I guess, for a few years, but I'd love if you could, make a little bit of an introduction about yourself and, basically what was your path from, you know, from where you started to, working on longevity first, and then founding, Ripper Bio, and then working on, really exciting, I guess, therapies for cardiovascular disease.
Why don't you go first for a change, Ward?
Yeah, sure. So, I'm Murat Topers. I'm the Chief Scientific Officer at Repair Biotechnologies, and I'm not a co-founder of the company. I should mention that. I joined the company back in 2020, right when COVID hit. Prior to that, I was a principal investigator at the Harvard Medical School for about a decade. During this time, I was also a group leader, team leader for a drug development program at Pfizer. And so my formal training, I started this at McGill University in Montreal, Canada, where I did experimental medicine. This is a doctoral research program. And after that, I pursued postdoctoral training at the University of Toronto in the Department of Immunology. And then, I was quite lucky. I got some good papers, good publications, which were very hot in the field. And then, I was recruited to Harvard Medical School, where I became a PI for about 10 years. So I kind of went in a roundabout way. But really, my training is very sort of much more hardcore, basic science, but translational to disease. And in particular, cardiometabolic disease, how the immune system interacts with that, and how the vasculature adapts in cardiometabolic diseases. And so, my meeting with Reason and Bill, the co-founders of Repair Biotechnologies, was really spearheaded by my friend and colleague Richard Honkinen, who also happens to be the inventor of the technology that we use at Repair Biotechnologies. Um, and so he introduced me to Reason and Bill in 2020, and really we hit it off. At the time, I wanted to transition away from consulting and doing basic science research into more, being involved in a company, because consulting for big, large pharma companies or medium-sized biotech companies is great, but you're really not involved after you give them all the ideas or you give them the right direction. You kind of lose, you lose that contact because things become much more confidential, and you cannot pursue that line of thought anymore. So it was refreshing to me to meet both Reason and Bill. And when I looked at the technology that we were going to use, I really felt that there was something really powerful there. And so I'll stop there, and I'll let Reason introduce himself.
Yeah, so I'm Reason. I have a background, I mean, primarily for this point of view, in the patient advocacy space. I have been advocating for more funding, more progress in aging-related research and development for quite some time now, since the turn of the century or so, though early efforts were rather crude. I helped out with the SENS Research Foundation and Methuselah Foundation somewhat back in the day, helped them raise some funds here and there. And just generally kept some websites going, Fight Aging and the one before it that's now defunct and rolled in. But that basically let me, at some point in time, if you write on a topic consistently, everybody who's active in the space will email you and ask you a question. So I ended up knowing a lot of people, at least that way. And then when there was an industry to speak of, I did a little angel investing, helped corral one of the early sort of collections of angels from people who were previously donating to research, and didn't have to do that for very long because the whole thing kind of took off. And then after that, I met my co-founder, Bill, and we founded a. He persuaded me that since I knew everybody, raising funds in a rising tide would be easy, or at least possible. So it definitely turned out to be possible, and I wouldn't say it was easy. And we started Repair to just go off and do some things that we thought were interesting. And along the way, one of them was the program that kind of took over the company and kicked out everything else we were doing, which is this way to break down cholesterol that Richard Honkanen, Morad's friend, put in play. And, he was a connection through Aubrey de Grey at the SENS Research Foundation back when, because Aubrey makes a habit of just finding obscure research and connecting with researchers, and he found it. And Richard Honkanen presented at one of the SENS conferences, and then connected to me. And I took one look at this technology and said, we have to do this. Fortunately, it seems I was right. So the first studies were amazing, and then we pulled in Morad via Richard Honkanen when it became clear that we needed adult supervision, and well, that's kind of been going great since then.
Cool. So yeah, I mean, I like to recall to fight aging, which I read also for many years, and I think it's a fantastic resource. Lots and lots of really interesting publications. I mean, if nothing else, I think the annual reports on what has happened in the world of longevity are fantastic, and a great summary for even for people that are not experts. It's maybe a little bit too technical for most people that are not familiar with longevity, but it was fantastic. But what was a little bit of what brought you to longevity? What was the thing that you woke up one day and decided, okay, well, why are humans dying at 80 instead of 100, or what was the kind of reasoning process behind that?
Well, I'd love to say I was a rational person, but I did literally wake up one day and decide that I really didn't want to die. So that was early stage me. Later stage me, I'm a little more copacetic about the realities of the world. You don't have to like them, but you get used to them. But I'm still interested in trying to make a dent in this problem before I can't do things to make a dent in it anymore.
I think for me it was when I realized when I woke up in the morning and I didn't feel as vibrant and as powerful as I wanted to be. So the aches and pains, and I realized that what when the aging process has started, I want to make sure that I age as slowly as possible, and if we could extend our wellness beyond what we can right now, I'm all up for it. Of course, I should also add that I was not really focused in this on aging. I knew that this was a problem, but really speaking to reason, the writer and the founder of Fight Aging, of course, it's contagious. His enthusiasm and his dedication to the field is contagious. So I should say that he directed me into this as well.
Yeah, I mean, I think of course, now you're, speaking among, people that, I think we're all, very much, of the same opinion, right? But, if somebody else is listening to this, and then maybe they're not really as convinced that we can actually do something about, reducing the problems of aging or whatnot, is there one, or a couple of quick things that you would say to maybe convince other people to start looking into the field or learn a little more about it, or kind of, start doing their own research and try to see if, or, maybe convince themselves that this is actually an area where we can make a meaningful difference.
There was a very interesting study a few years back in which they demonstrated that 10% is the magic tipping number. You don't need to convince everybody, but if you can convince 10% of the people that it's inevitable that whatever you're doing is going to become the mainstream at some point. So honestly, I don't try very hard. You just put the number out there. You just put the details out there, and if somebody's interested, they're interested. If they're not, my experience is, there's nothing you can really do in the short term to persuade them. It's a many touches type game. This stuff has to be out there. People have to be talking about it. The person has to come back and see it from different directions at different times and slowly start to feel, that is the thing. Most people are not looking to be convinced, in the sense of, here's an argument, this argument is correct. They're looking to be convinced in the sense that, oh, all these other people think that this is right. Um, and so you have to win over the people who can be argued with and convinced rationally, and then they prefer serve as social proof for the rest. That's my take.
That's absolutely true, and I agree with that. But I also think that more and more people want um, clinical validation. They don't want anecdotal evidence. There's a lot of saying, well, I take magnesium glycinate. It helps my mental acuity, and if I take this, I feel better. I sleep better. I feel younger. You could say all these anecdotal sort of the way you feel. But I really think that the field would really advance if there was some form of clinical validation showing that there is actual data out there. And like, if there is any, if you're stopping telomere shortening, well, then you know what, there is clinical evidence for that. If you're affecting other hallmarks of aging, there's clinical evidence for that. And I think that's when people are really going to start coming, uh, you know, coming to terms that aging, there is something that we can do, and there is a light at the end of the tunnel. So if I follow a certain regimen, I'm going to get to where I want to be. Whereas now it's harder to convince people, and people just have to come by themselves and say, "Look, I really want to do something about it."
There's a hierarchy here. You got to convince some scientists, because scientists are the people who produce new technologies, at the head of the spear. Then, having got some technologies, you have to convince the physicians, you have to convince the regulators at the same point in time as you're convincing the physicians, because some of these things can be used by physicians prescribed right now. Some of them need to go through regulatory processes. But either way, the goal is to get a big enough weight of data, a big enough set of people convinced. And once physicians are, sold on something, then everybody else just does what they're told, because the physicians very much steer what goes on in medicine. So you have this sort of, right now we're in absolutely the earliest stage of trying to convince the world, which, as Morad says, you got to get the data, you got to have the clinical trials, which other people will say, oh, that makes sense, that this must be right. And then let's go use it. Let's go introduce it into clinical practice, and patient advocates groups will start asking for it. But until you have that data, it's very challenging. And one of the biggest problems in the field right now is that while we certainly have technologies that I believe are probably, as effective as exercise, perhaps more effective than exercise to some degree. I mean, mTOR inhibition, senolytics, fecal microbiota transplantation or similar. These things are all very cheap. Anybody can do it. Physicians are prescribing it, and there is next to zero human data for any of this, because nobody has any incentive to run the clinical trials at cost. I mean, if a philanthropist could spend $10 million and answer all the questions I wanted answered here, but nobody's lining up to do this yet.
One thing that I would add is, completely agree with you that um, there has to be some emotional basis to it, right? Like, it's not just a matter of a rational decision process. There's this quote that I really like, which is, uh, ideas that come from emotion don't go away with facts, right? If people just do not feel that longevity is possible, right, or extending longevity is possible or whatnot, no amount of data would convince, I think, the vast majority of people, right? So what you say about, maybe just convincing, that tipping point that can actually make a meaningful difference, right? Or the people that can be convinced with facts and with data, maybe that would be the only option. I mean, I was thinking, it would be, uh, it's interesting when you look at, for example, exercise, right? Like, we've known that exercise is good for people for decades, right? And how did the I guess public health effort of convincing people to do exercise have worked out? Right? I mean, I don't know. I don't.
I mean, one could say the same thing about eating healthy, right? I mean, people know that, there are unhealthy foods and healthy foods. And I mean, exercise might be because you have to make time for it, but everybody has to eat. But the fact that people just keep on making bad choices when they're eating, I mean, that bar is even lower, and people still need to have that sort of aha moment and say, you know what, I'm really going to make a change in my life because I want to live better, longer, and be more fit. So I'm going to change my diet, right?
And I think
So
the size of the effect matters. If you have a very large effect size, a very rapid effect size, people are going to be a lot more motivated to do something that's essentially preventative, um, because they see effects. I mean, look at and the other thing we might mention is statins, and statin compliance. Statins like don't have a very large effect size in terms of your mortality. They have measurable effects in the short term on things that don't change the way you feel at all. They're just blood parameters. But in the long term, it's a 10 to 20%. If you believe the consensus studies, and a lot of people just aren't. They can't, get out of bed to take a low cost drug, 10 to 15, 10 to 20% of effect size on mortality. And you need something bigger and better than that if you want people to jump up and get on the prevention train.
Yeah, one interesting thing about the sensory effects, I was talking with somebody recently. Um, it seems like a lot of the, I guess, lifestyle habits, good nutrition and exercise and getting enough sleep and so on, they seem to make a meaningful difference in average lifespan because they kind of weed out, people that maybe, would die early because of very unhealthy habits or alcoholism or heart attacks that they get when they're young or whatever, right? But it doesn't necessarily make a meaningful difference in maximum lifespan. It's not like we have Olympic athletes, living to 150 years, right? It's not like, we have, like Instagram influencers that seem to, count every calorie, living meaningfully, longer, right, than average people. So, yeah, I wonder, what are your thoughts on that, right? Like, why it seems to make a difference in, you know, I would say, kind of removing those unhealthy habits and dying early, but not necessarily meaningfully extending maximum longevity.
Well, there's an interesting problem. I don't think anybody really understands why compression of morbidity exists, right? How can you, at a priori, if aging was really like reliability theory, the reliability theory says, where okay, you're a big lump of many 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 redundant parts, and redundant parts are either working or broken, and that's what the model produces things that look very like human aging. It's an interesting field of study. But clearly that's not the case, because reliability theory would predict that if you can slow aging, you're basically extending everything. You extend the period of health, you extend the period of disability. You're just slowing everything down. But that's clearly not what happens in the real world. And there's ways in which, so it's not that simple. There's some way in which the multiple processes of aging interact, which say, okay, we can address some of them, but then some of them we're not doing so well on. And any one of them can kill you. And for many years, my simple model here is that upregulation of autophagy can slow a lot of things down connected to inflammation and mitochondrial function and senescent cell burden, etc. But it's not doing very well with, let's say, transthyretin amyloid. And maybe it's not transthyretin amyloid, maybe it's something else, but there's something, possibly several somethings, that are acting as sort of the full stop at the end of the sentence, and that nothing you can do with lifestyle or exercise really influences them all that much, which is how you end up with the ability to push out your healthy longevity, but to really struggle with pushing out actual lifespan to the same degree through what we can currently do with lifestyle. And this is a model. This is just a way of thinking about things. I have no idea whether it's true or not, and it's fairly likely that we're not going to find out whether it's true or not for a very long time, because people, if you think about what can be done with aging is going to be a treated concern. There's going to be serious rejuvenation, actual large amounts of rejuvenation for decades before people have any idea what what the relative effect sizes are on human mortality for all of the various options. And that's just the way of it, because if you look at, like, let's say statins are not a settled discussion, right? That 10 to 20%, that's the consensus voters. But there's plenty of people who say it's zero. Um,
I think
and
there was some paper recently showing that they may kind of, end up increasing the chances of dementia as well.
yeah, same with aspirin. Aspirin is like an ongoing argument where people are very heated about, is it actually good or not? What's the effect size? And if we can't, after 100 years, have a good idea about what aspirin does to human longevity, I'm not optimistic that we're gonna have really great data for what senolytics do to human longevity anytime in the next few decades.
But I think we could also be, hitting the maximum of our based on our genetics, right? You're by doing these interventions, what you're doing is you're providing us a scenario where it's the perfect setting to live what your genetics allow you to do. And that epigenetic reprogramming is really going to start extending life beyond what we know today. Because epigenetic programming could have different multiple genes being affected at the same time and a cascade of pathways being affected at the same time. I think that's what you really want to do if you really want to extend life beyond, say, our centenarian sort of limits, which is the best case scenario. Like, for instance, if you take statins, right? If you're not going to die from heart disease, cancer is going to kill you. If you're not going to, there's other diseases that are going to pop up, or dementia is going to get you if you're not going to die from cardiovascular disease, right? Cardiovascular disease usually affects individuals between the ages of 50 and 70, and they either go into a spiral and they die over the next 20 years, or they treat themselves with statins, with careful monitoring, and they kind of extend their lives to their genetic maximum. But if we really want to have a big impact in terms of longevity, we really need to look at epigenetic reprogramming. I mean, look at the queen bee, right? The queen bee, I mean, they're all, the beehive is genetically identical, but that jelly that the queen bee consumes makes it 3 times larger than other bees, and also has a much longer lifespan. This is all due to epigenetic reprogramming. So if we can adapt the same sort of principles to humans, then I think we could really see a big impact on longevity and wellness.
Yeah. I mean, I think back when I started learning about longevity, right? I remember listening to Aubrey de Grey, right? He was talking about, uh, well, let's think of humans as cars, and if you maintain the car will last longer, right? And that, very much appealed to me, right? Like, this perspective of, like, a lot of aging is caused by damage. If you repair the damage or prevent the damage, you could arguably extend longevity. And I think purely that damage repair approach makes a lot of sense, and I really like. But I think the more I learn, the more I realize that maybe that's not all there is to it, right? Like, maybe there is some genetically predetermined factors, right? Like, that are not just related to the damage, I guess, and maybe that's the reason why extending maximum longevity is a lot harder than extending the average. I don't know. I mean, I'd love to hear your thoughts on, like I said, all damage related, or there are other things that we may still don't understand completely.
Well, I rather like the current work that came out on the DNA double strand breaks producing characteristic epigenetic changes of aging. It still needs some more validation and work on that, but it would certainly explain a lot, if that's the case. The reason that some underlying process is causing the epigenetic changes that produce dysfunction characteristic of aging, and it's a damage process. It's not like it's a process that can't be evolved out of, because we need a certain amount of mutational damage for evolution to work. So you would that would really solidify the damage view of aging, to my mind. As I see it, if you're intervening on something other than repair, then I feel you have a pretty steep hill to climb to demonstrate that what you're doing is sane and is actually going to move the needle. I mean, there are obviously things that you can do that are not damage repair that have meaningfully large effects, such as I mean, technically clearing senescent cells isn't addressing the root cause of the burden of senescent cells, but looks pretty good. Similarly, control of blood pressure doesn't address the root causes of why your blood pressure is screwed up, but it's also, a 20% mortality reduction with cheap drugs. So, there are obviously targets one can go in there and address without dealing with root causes, still get something beneficial out of it. But I've always felt that if your goal is to mess with metabolism, if you're trying to build a better operation of metabolism, that's a far future concern in comparison to what we can do today, which is hopefully a little bit of damage repair. But making new genomes and changing the one we have to be better, this is a really hard problem. And you see that in the comparative biology of aging, where people have been studying naked mole rats energetically for more than 20 years now. And I think last year was the first year that somebody took a naked mole rat gene and put it into a mouse in the hopes of producing some beneficial effect. And it didn't work so well. So, we're a long way from taking things that we learn about in other species and trying to introduce them beneficially into our own genome. Not least of which because gene therapy in adults to produce sweeping changes is not possible and may always be very hard. And just changing the next generation, good for the next generation, but it doesn't solve all problems.
Of course. Um, yeah, I mean, I think this is one of the problems that everybody kind of mentions, right? Like, even if we could make gene therapies work effectively, right? Like, the distribution problem, how to get every single cell in our body change. Uh, this seems, still very hard. And I don't know if anybody has a, great idea about how to actually solve the problem.
Well, they are changing this with CRISPR gene editing, right? They're doing base editing right now, or CRISPR editing, and even if they're changing a smaller number of say uh, the genomes of correcting the defects in a few cells, which basically, let's say it's a receptor or it's a secreted protein that is providing potentially cures for some patients, right? Some patients that are defective, let's say, for instance, the LDL receptor. There are companies such as Verve Therapeutics right now that is working on correcting the LDL receptor and providing some relief for these patients. So the field is moving towards gene editing and correcting already known genes. But the key here is to identify master regulators of pathways. And we're not there yet. Not all the pathways are active at the same time. I mean, that would be energetically impossible, right? The cell would just exhaust all of its ATP. But there are certain pathways that are more dominant than others. And we just haven't really figured out which ones are the dominant pathways to target for aging versus others that are just ancillary, which are the backup pathways. And so that's the problem here. When you want to do gene therapy, which pathways are you going to target? Which genes are you going to target? And are they going to start a cascade? As Riza mentioned, we're very, very far away from that, perhaps decades.
Yeah. You mentioned another thing, before, which is, well, cardiovascular disease, tends to affect people, I guess, 50 to 70. And then if you don't die of that, then maybe you'll die of something else, right? But I guess there's one very interesting thing, which is, well, can we look at the causes of death of people, right? I guess cardiovascular disease is the largest one by far, and then solve those one by one. And then I mean, I think sounds very reasonable that if you could solve enough of those, right, like you could, push the maximum, longevity, right? And this is the thing that I think is, maybe very exciting about what you're doing at Repair Bio, right? Like you're not just trying to manipulate some of the markers, right? Like lowering, you know, uh, LDL in blood, right? But actually solving the underlying problem, right? Which is this, plaques that form in the arteries, right? So maybe you can talk a little bit about that, like, what is the rationale, like, how does this technology work, right? Like, it was maybe the kind of the ideas that led to it versus, say, working on, a better statin, right?
Yeah, Mora, do you want to take that?
Yeah, sure. So this was really the brainchild of Richard Hawking. He's a close friend of mine. He actually tried to recruit me to the University of South Alabama back in 2014. And I went there. I spent about a month there, just to survey the area, and I ended up coming back to Boston, but we remained friends since. And Richard is an avid sailor. Okay, he loves sailing. He loves boats. I should mention. And during the 2010, there was a Gulf oil spill, where the Gulf of Mexico was basically flooded with oil, and he was thinking about can there be ways to actually degrade oil, petroleum. Now, this is, of course, far away from cholesterol, but I'm getting to that. And because he's in the Department of Medicine at the University of South Alabama, he wanted to write a grant. So he said, what if we can degrade cholesterol? So it turns out that tuberculosis, a bacterium, actually lives in the endosome in the lysosome of bacteria of macrophages, and the way it remains dormant that way is because it actually feeds on the cholesterol inside the lysosomes. Right? It could actually use that as a carbon backbone, carbon source to sustain itself until the immune system of the host becomes weakened, and then of course it gets activated, and then it could wreak havoc in the lungs of patients. Um, so he was looking at that, and then he started getting the idea. Well, there is precedence that you could degrade cholesterol. Can we take a few proteins that are present in our bodies, and can we combine them in a way that we could, put this fusion protein in humans and cause it to degrade excess cholesterol? And that's exactly what he did. And reason, as he mentioned, he saw this technology and he said, "Look, we really have to put this into humans." And that's how we began our quest to optimize this technology. We've optimized it maybe now eightfold or tenfold, and brought it into a form where we could actually use it therapeutically. Do you want to continue on, Reason? I could pass it on to you from here.
Yeah. So, I mean, we tried it with normal, the everyday gene therapy techniques, say like AAV, as the outset. It worked great, and we migrated across a number of approaches to therapy because AAV high dose systemic into the body is sort of out of favor at this point in time. There've been a number of deaths and other adverse reactions in clinical trials in recent years, and while, the FDA remains open to it, it's certainly investors do not, to anywhere near the same degree. It's a hard ask. So we walked off and looked at cell therapy approaches, deliver macrophages engineered to be able to break down cholesterol, into the body. And you can actually, if you take a mouse that has 1 million macrophages running around its circulation, you can give it 10 million macrophages by injection. And the mouse is fine. That doesn't do any harm. So in principle, you could throw enough macrophages into the body to actually go and attack plaque. But one of the things we learned in the course of doing this is that cell therapy is enormously expensive and complicated. And while at the time we were doing this, there were some very big players who were looking great and making waves. Things are not going so well for them now. A lot of them have burned through an awful lot of capital without getting to where they want to, and orders of magnitude more capital than we had access to. So it's just not a good idea unless you had $1 billion sitting there to pursue novel forms of cell therapy. Cells are obnoxious little monsters. They will do what they want to, and they're very hard. Never work with children, animals, or cells. Is the errors. We eventually moved away from that to this all happened during COVID. But by the end of COVID, everybody understood that the formerly sort of marginal lipid nanoparticle mRNA mode of therapy was actually really great. And now everybody understood how it worked, the up and the down. There was manufacturing that was established. All of the parties involved in this were much higher profile. So we gave that a shot. And it's kind of the least worst option, really. I mean, the problem with gene therapy delivery is it is hard. And if AAV is not available anymore for anything other than local local injections, then you're a little bit stuck with you either develop something new or you're using something that's very tailored to a very niche use case, or your lipid nanoparticle mRNA. Those are the only options on the table right now. And there are a lot of people trying to produce new options, and we've spoken to a number of them. And honestly, a lot of them are just not ready. They're not ready for prime time. They're not guaranteed to work for you. If you have 5 years, then it's great idea. Go out there and like play the field, see what's out there. There's a lot of really fascinating technology. There's a group out there that actually has a technology that makes use of the chylomicron, which is a particle that transports material that you eat. So they have essentially an orally available lipid nanoparticle type affair, and things like this. There's a lot of very fascinating modes of delivery that out there. But they all have their pluses and their minuses. But if you're at the stage that we were, then you simply have to use something that just works, is proven, is as close to being nothing new as possible, and forge ahead with that. Because otherwise, you're looking at several years of trying to get something to work if there isn't already clinical proof. And
Why,
right.
uh, why is it hard? Like, what are the problems that you find? Is it because the body reacts? There's some image
It's not.
to it.
It's a lot. There's a lot involved, right? For the lipid nanopart. Well, for the AAV field, um, s- high dose systemic administration of AAV is very dangerous. I mean, there was just a recent death that occurred in a young, otherwise, I mean, they had a genetic defect, but otherwise healthy individual, where high dose AAV systemic injections can cause it could be fatal. But in terms of the lipid nanoparticle field, one might think that it's very, very simple. It's very straightforward. You just basically mix a lot of lipids together and you encapsulate mRNA. But there's a lot of IP involved here, and there's a lot of what what you don't know might hurt you. Meaning that you might develop something which is basically owned by someone else, and so in the sense in the case of lipid nanoparticles, it's better to collaborate with the inventors and the leaders in that field rather than just developing it yourself. And that's what we've done. We've actually formed a very strong collaboration with Genevant Sciences, and these guys are the 800-pound gorilla in the room. They have all the patents, almost all the patents, and so therefore you have that freedom to operate when you assign a deal with them. So that was our whole. I should maybe add that we decided on lipid nanoparticle technology to deliver mRNA because everything just connected. Meaning that with COVID and bringing lipid nanoparticles into the medical field, more research was done on it. Better products were developed, and we happened to be there and say, well, look, we could use mRNA delivery rather than using AAV, which was a big benefit because putting AAV, especially in patients where they have compromised livers, is a no-go. You will never even get approval by the FDA for this because the chance of having a fatality is that much greater, right? So that's why
Well,
we
I mean,
said,
from a technical perspective, right? Is the problem that these sticks are hard to manufacture, or that they don't work when they are injected, or that they don't go where they're supposed to go, or that the DNA just doesn't stick there, or that it kind of like what are the like, if somebody starts from scratch and says, well, let me go to this say manufacturer of nanoparticles that you have, I have an idea for a potential therapy, right? Why would it take 3 years instead of 3 months?
Yeah. So the problem is it's complicated. And there's a lot of work that goes into taking even something that is very well controlled and getting it into the GMP production that's needed. You need to be very capable of controlling and reliably producing things. And when they're so complicated, it just takes a lot of time to do that because nothing ever works right the first time in biotechnology ever. There's a lot of things you have to constrain down, and simply, I mean, in research, most papers don't reproduce. Most people's claims about the technology they're developing don't reproduce. Most of these things that people develop are nowhere near as good as they think it is, and they're working in one particular constrained system, and you come in and put their stuff into some completely different system and context, and odds are good it's just not going to work. We've seen that, and then why doesn't it work? Well, who knows? It just didn't work. And now you have the problem of, okay, how do we debug a piece of biotech? And that's an obnoxious exercise in and of itself, because now you're essentially wolf fencing. You're trying to like find specific areas where you can identify what might be going wrong, and you have to conduct dozens of studies, and it's a very slow and painful process when something is not reliable. And most things are not reliable until they've had a great deal of work done on them. That's really the problem. It's a problem of complexity and inadequate tools.
And I should also add that, site-directed delivery, like specific organ-targeted delivery of lipid nanoparticles, we're just not there yet, right? When people could talk about this, but really the main organ where we could deliver lipid nanoparticles with a lot of assurance is really the liver. And the reason for that is that the liver, like other glands, they have a certain type of endothelium. This is called fenestrated endothelium. So the endothelium in glands actually has holes in them. They're fenestrated. And these holes are approximately 200, 250 nanometers in diameter. And so when we use a lipid nanoparticle that is of 80 nanometers in diameter, it could freely cross these fenestrae and then directly interact with the cells of that organ, of that gland. And in this case, the liver. And because most of the blood flow, a lot of the blood flow goes to the liver, the first site that these lipid nanoparticles go to is the liver. In addition to that, our lipid nanoparticle has a specific ligand on the surface of the lipid nanoparticle that interacts with a receptor that's only expressed on hepatocytes. Now, we are lucky that by delivering to the liver, our enzyme is able to degrade this excess free cholesterol and regress atherosclerotic plaque. But to say that lipid nanoparticles are at a stage where you could deliver to any organ and have salutary effects, we're not there yet. And there's a lot of research and development that's going on right now, but we're just not there to say that a particular lipid nanoparticle will be robustly expressed, like say, for instance, like the heart. We're not there yet. So a lot of work has to be done.
And then the theory is that, or I guess that what you have seen is that by delivering these lipid nanoparticles in the liver, then we just mRNA, right, encapsulate it, right, and then they express this enzyme that can degrade cholesterol, and it degrades cholesterol in the liver, right? But then, how does that kind of remove the plaque that may already exist? Like, is that understood, or like?
I mean, I'd like to say that we understand it, but there's a lot more work to be done. And I think we're just scratching the surface. We are affecting multiple pathways at the same time, whether it's the immune system, we're affecting the lipid transport mechanisms, and I think that we are affecting other mechanisms that are yet to be unraveled. But what we have found is that we are getting profound immune suppression that would otherwise cause progression of atherosclerotic plaque. And it's also heightening the regression of plaque by transporting more of this free cholesterol from the plaque back to the liver where it gets degraded. But of course, we believe that there are other mechanisms at play. But we believe that our therapy is unique because it not only affects free cholesterol in the liver, but it also unleashes other protective mechanisms. We believe that humans have these mechanisms to regress plaque, but it's just that with age, what happens is that these mechanisms become downregulated, and therefore we deposit more plaque rather than regressing it. And by just healing the liver, we are now reviving these innate mechanisms that we have to regress plaque. Reason, would you like to add to that?
Yeah, I mean, I think the thing that we take away from our work, I mean, obviously we're at the frontier here. We can't point to anybody's research to say, hey, this is how it works, because nobody's targeted free cholesterol before. It's an undruggable target. It's sitting there inside the cell, and of course the cell's membrane is full of cholesterol, and there's a bunch of cholesterol in droplets that's esterified, and you can't just run in and do things that would get rid of cholesterol, because you'll kill the cells. You have to very carefully target this specific excess free cholesterol. So nobody's done any work on this really. The liver community are the closest to having some body of literature that says we think excess cholesterol is bad, but we can't test this because there's no way to get rid of it in isolation of causing other changes. But nobody else has really put a lot of thought into that. I think the neurodegenerative community is starting to come around to the idea that lipid dysfunction is a thing, and lipid droplets are actually important where they occur in the brain. And particularly the Alzheimer's community is starting to notice that the ApoE4 variant is actually causing lipid metabolism dysfunction in microglia that connects to inflammation, et cetera, et cetera. Anyway, so the 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 upshot of all of this is that you have to be able to clear out you have to be able to clear out the free cholesterol in a way that gets rid of it permanently. People then, and the way we do it is sort of poking at the liver, which is the linchpin of the cholesterol transport in the body. It's the central point of which regulates all of these things. So if you can just put a cholesterol deficit in the liver, then you're going to get all the cholesterol in the body flooding that way through reverse cholesterol transport. Now, people have tried upregulating reverse cholesterol transport in the past in isolation. You literally just put more HDL particles in a person or some other approach. But it doesn't work in people. They tried it. And our thesis on why that doesn't work in people is that what you're doing is you're moving excess toxic free cholesterol from the rest of the body back to the liver, and you're not getting rid of it from the liver. It just sits there and screws up the liver. So that absolutely abrogates any possible benefit you might cause, because now the liver is screwing up the rest of the body. So you have to transport material back to the liver. You have to break it down when it gets to the liver. And the other thing that our therapy does is cause a dramatic reprogramming of the immune system to significantly depress inflammation. So I think you have to have these three things. In order to produce dramatic regression of atherosclerotic plaque after the way we're doing it in animal models, you have to have these three things: reduced inflammation, upregulated reverse cholesterol transport, taking cholesterol back from the rest of the body to the liver, and break it down when it gets to the liver to stop it messing up the liver. You need those three things. This is our thesis. Now, obviously, we can't point to papers. We can't have somebody else come in and say, "Oh, well, yeah, of course, that's how it works," because we're producing data that nobody's produced before, and we're just trying to fit it in with the established literature to understand, what could possibly be happening here reasonably, rationally. What is the medium? What is the sensible way in which we copy producing these results? And we generate data, a lot of data, in an ongoing fashion. Murad, if you want to.
Yeah, right. That's perfectly said. And when we bring this up to key opinion leaders in the field or other experts in cardiovascular disease, and everybody says, "Oh yeah, that makes a lot of sense." But until now, we couldn't have tested this because, as Reason mentioned, this was an undruggable target. You just can't target free cholesterol without killing the cell. So you really have to target only the excess free cholesterol and not the essential cholesterol. I mean, for your reviewers, cholesterol is vital for cellular function. You need it for steroid synthesis. You need it for membrane fluidity or rigidity, I should say. You need it for bile salt production. There are many pathways where cholesterol is involved. So just by putting an enzyme in a cell and hoping that you're going to degrade the excess free cholesterol, you might kill it. So what we did was we engineered the protein in such a way that it only degrades the excess free cholesterol, and this has huge salutary effects, as we've seen.
You couldn't like come up with a small molecule that binds to it, right? Because it may bind to the one in the membrane, it removes some
Right.
and then the cell kind of breaks down.
Exactly. It's a fine tipping point, right, for that. So we had to be very careful.
And maybe to close the loop with longevity, right? Like, so you're starting this with people that have familial hypercholesterolemia, right? Just like a genetic disease in which they have a lot more cholesterol than people normally have. But I guess the idea is, if this treatment, is approved and it proves to be very, very safe, then a normal person, could take it as well, right? With the notion that by reducing the chances of having these plaques, you probably would eliminate the risk of having cardiovascular disease or strokes or heart attacks and whatnot, right? So like, I guess like you wouldn't necessarily have to prove that it extends longevity, right? But the point is, like, if you remove the chance of dying of a heart attack or a stroke, right? Like, the person would live longer, right? Is that, did I get that right?
Yeah, I think the physicians are fully on board with this whole idea that that getting rid of plaque is a good idea. Interestingly, we live in a world in which the regulators do not think that getting rid of plaque is a good idea because nobody's produced or reversed, a randomized controlled trial in which you show that getting rid of plaque is a good idea because there's no way to get rid of plaque reliably. So nobody's been able to do this trial. So you have this catch-22 situation in which you can't the regulators won't let you approve a drug for getting rid of plaque. So along the way to getting your drug approved, you have to show that it does something else, such as improve exercise tolerance or, reduce liver dysfunction or, anything, or indeed reduces heart attack and stroke, except that that's going to be a very long and expensive exercise. But you kind of have to, it, the regulators are the only people who are taking the position that no good comes of removing plaque because nobody's demonstrated that any good comes of removing plaque. Everybody else understands that removing plaque is the holy grail.
Right. But we do believe that with age, we accumulate this free cholesterol in the liver, which causes impairment in these innate mechanisms that regress atherosclerotic plaque. And so, as you age, you have more and more free cholesterol that accumulates in your liver and other parts of your body. So by getting this therapy, we not only regress your existing plaque, but, we have this added benefit. We have seen that in our mouse models, the animals increased exercise fitness. They had increased exercise tolerance. The mice that can go on a treadmill almost twice as long until fatigue versus animals that are not treated with that therapy. So we believe that there's both an improvement in liver metabolism of energy, and or energy utilization, as well as a better functioning vasculature for better blood flow. So this could benefit, as you mentioned, an aging person could actually take this and be revived, and which would then, of course, feed into them exercising more, living a healthier life, and this is basically a snowball effect that we foresee.
Yeah. So excess intracellular cholesterol should probably be a hallmark of aging, but it's not our crusade to have that added to that list because it doesn't help us in the slightest to have that, to spend that time doing that versus other things we should be working on. If you've ever wondered why companies developing, putatively anti-aging drugs don't run lifespan studies, it's because investors don't care. And investors care if you do stuff with their money that's not directly going to get them their payday by getting an approved drug, right? And lifespan studies are just not one of those things. But I'm willing to bet that we would extend the lives of these mice if we actually ran that study because we are tremendously improving their health.
Yeah, I mean, but I don't know if we wanna open that kind of worms, right? But like, why would you say investors or people in general are so irrational in that sense, right? Like, you see folks that, maybe get to like 80 and then they're sick and then they say, oh, I wish I could live a little bit longer. I would, give up all my money, if I could get a few more years of life. And of course that's false, right? It's like, people say things, but they don't really believe it, right? Like, what, I mean, I don't know, like we could speculate, right? Like, what could be in people's heads, right? Why send them more effort on longevity, more people jumping into the field, more money being deployed, right? Like, what are, I don't know, maybe some of the things that can be done apart from, advocacy, right? Or just,
Well, yeah, there's a ruthless sort of evolutionary fight going on in the investor world, and at any point in time, you should expect that 99% of the investors that you encounter are people who have optimized ruthlessly for making returns and only caring about having capital because everybody else is gone. They spent their capital. They went and did something good with it, perhaps, but that capital isn't there to go invest in things in anymore. So the ones that are left are the ones that really don't care about anything other than ROI, and they will be anything and everything uh, to go get that ROI, including the usual diverting biotechs from their intended goal to go work on obesity, for example, which is what is happening to the earliest wave of companies in the longevity space.
Yeah. Um, I mean, for the good or for the bad, right? Like, you could, maybe like, so say, well, if somebody else in longevity is able to maybe turn a little bit of the attention, right? Like, to bring more attention into the space, right? Even if it's under the sky. So like making people skinny, right? Like, that could be welcome. And I mean, I don't take Ozempic or anything like that, right? But I guess, the people that take it, would see, they would probably, many of them would be healthier, right? Like, and then that may, apparently there's some studies saying that, it may actually extend longevity as well, right? So, I guess it's, overall positive for the field, even if it doesn't kind of, follow the routes.
I guess for obesity per se, it's a visual feedback, right? You see that you are looking better. You fit into your clothes that you used to fit into 20 years ago, for instance, right? So people are enticed by this. They see their friends, they lose weight. But for cardiovascular disease, it's a silent killer. Like, the adherence rate to statins, as a reason we was talking about at the outset, it's very low. People will not take statins despite the fact that their physicians tell them, if you don't take this, your plaque is going to burst and you might die. So that we need that visual, that sort of, we have very short attention spans when it comes to results. We want results today. I think that's a big barrier. If people do not want to plan ahead, people, what does it say? They say that people always overestimate what could be accomplished in a year, but underestimate what could be accomplished in 10 years, right? And so we're very short-sighted when it comes to results. And if we don't see something quickly, we lose interest. We don't want to do it. We don't want to adhere to it. And I think the same thing goes for investors. They want to see results right away. Are people going to be interested in this? And if they're not going to fund it. They don't want to put money into that.
We need more philanthropists
Yeah.
to short circuit the existing financial incentives, but keep people away from doing the sensible things and working on stuff that will actually make a difference, but not to somebody's bottom line, but indirectly to making the world a better place. And to their credit, a lot of philanthropists threw a bunch of money at this problem in the last 10 years, and that's kind of why we have an industry, because there were a lot of people, high net worth, putting money into this and being enthusiastic. But they have short-term horizons, and they all learn that biotech is a very slow game. But you need some true believers, and somewhere out there are going to be true believers who are more true believers than the current true believers, because they will actually spend down their money to make things happen, rather than the people who are putting some money into it to make things.
We should also thank our investors. We're very grateful for the unwavering support. The high net worth individuals, and even the not so high net worth individuals that have supported us along the way. I mean, we're very grateful, and it allows us to bring this therapy to the clinic. And so we can't thank
Yep.
them enough.
This would never have happened our company would never have happened in a normal market. No investor would have touched it. No normal investor would have touched this. Normal institutional investor would have touched this with a 10-foot barge pole. It's just not their thing.
And it's a shame. It's a shame because it's a great technology, right?
Yeah, I mean, and of course, there are many more, right? Like that do not even get that initial funding, or they don't get enough exploration. I mean, you mentioned the funding. You talk a little bit of the regulatory aspects. You will also talk a little bit about maybe better tooling for, like, gene delivery mechanisms or whatnot. What else? What, like, maybe not what else, but like, what in your mind are the biggest obstacles to have longevity therapies out there, and for companies like yours to.
Yeah, I think that's back to the data. We need data, and the easiest data to get is the data that nobody's going to no for-profit concern is going to fund, because it involves off-label, off-patent drugs. It involves processes that you can't really patent, and that nobody's going to be able to protect, and they're going to be cheap. So there's no incentive to go spend 10 million or 100 million on trying to get an approval or produce large amounts of data, but we need philanthropists to come in and just fund trials of these things and actually prove, because I think we have the technologies that could produce a meaningfully large enough gain in lifespan and a meaningfully large enough reversal of aspects of aging in humans to change people's minds about, the value of this field. And that's senolytics, mTOR inhibitors, and something gut microbiome, whether it's Flagellin immunization or fecal microbiota transplantation. I think these things will have large enough effects to care about, is my belief based on the animal data and what we've seen elsewhere. Mm.
Well, um, with that, we're almost an hour into this conversation, which I keep saying it in every episode. It's about the maximum that you can get into a podcast, before, it starts forgetting what we talked about. It's about the time, that you will listen, commuting to work and back, right? For the, that go to work. But, maybe just, to answer, like, I don't know if there's anything else that we missed that you would like to share, or, anything that would be interesting. Maybe, also, like, people that may be interested in what repair is bio, like how they can or fight aging, right? Like, how they can, find you, and, anything else that you would like to share.
All fight aging and repair biotechnologies are both easily found online. Just use a search engine. It's a failing skill, I know, but they're out there.
Yeah, and we're always happy to chat about the technology and what our accomplishments are. It's just a question of reaching out to us if you want to learn more about our company, what our trajectory is, and how we're going about doing things, both on the regulatory front, on the scientific front. We're happy to share. We're happy to talk to you. And out here, I also would like to thank you. Also, these podcasts are important. To shed more light onto the industry, and we need to talk, and we need people to learn more about the longevity field, and it's very helpful. Thank you for this.
Yeah, yeah. No, I mean, thank both of you, right? Yeah, I think if one of these, podcasts, or myself or anybody else, right, ends up being like half as popular as, like, Lex Friedman or whatever, right? That'd be great. Like, maybe we should have more people talking about longevity therapy instead of, the latest, LLM du jour, or or other things that are, maybe not as important to people, actual lives.
Well, happy to talk to you.
Yeah.
Cool. Well, thank you so much. This is great. That's a conversation, and super insightful. So thank you for taking the time, and it was a pleasure having both of you here with me today.
Thank you, Amir.
Yeah.
All the best to you.